So welcome all to this IMSc popular lecture by Professor Smita Srivastava. As you all know, we have been celebrating the 60 years of completion of IMSc this year and also the Azadi Ka Amrit Mahotsav. As part of Azadi Ka Amrit Mahotsav, we have a various series of lectures we have been holding, and this is another popular lecture, and it gives me great pleasure in inviting Professor Smita Srivastava to give this lecture. Smita happens to be a friend. So that makes it very nice. So a bit about Smita and her group, and also the lecture I must mention is on a topic which will be highly applied, and you will get to see what happens in the neighborhood a bit. So Smita, for all of you, she did her master's and PhD from IIT Delhi, and subsequent to completion of her PhD, she has been a faculty in IIT Madras since 2010. Her research group focuses on high value plant phytochemicals. Many of them have therapeutic uses. I won't go into the detail of the bioprocesses she has developed. She will show you that as well. Apart from exciting research going on in these areas, Smita also happens to be the faculty in charge of the bioincubator in the IITM Research Park. Okay. So with that short introduction, which is common in IMSc, I invite Smita to tell us about that. I should mention that I requested Smita to have two parts of the talk. The first part of the talk, which will be about 40-45 minutes, covers her own research, and she would also introduce us towards the end of the talk towards various activities going on in the bioincubator of IITM Research Park. With that short introduction, Smita, over to you.
Thank you.
Thank you for accepting our invitation and looking forward to your lecture.
Thank you, Arijit.
Yeah.
So, thank you everybody. Good evening, and it's my pleasure to be here. It's for the first time that I'm visiting IMSc. I've been hearing about the institute but couldn't get a chance to visit. All thanks to Arijit to invite me here. Let's hope I do justice to it. Okay, so without any further delays, let me talk about what research we do in Plant Cell Technology Lab at IIT Madras. This lab is in the Department of Biotechnology, and when I joined the department, I had my previous research background in plant cell bioprocessing. So I really wanted, and nobody was doing this work in the department. So my objective was to come here and also encourage students to do this research and take this domain forward. So with that objective in mind, I could initiate the plant cell bioprocessing lab in the department, and then I would here be talking about what key research leads could come out from my wonderful group. Many of them are now in industry, and one of them is doing postdoc. So as a bioprocess engineer, we are always focused towards commercialization of processes. So how can we bring in economic and technical feasibility to these processes using biochemical engineering principles? So that's how I was trained. That's how I train my students as well. So even when the focus was on plant-derived molecules, the key idea was that how can we bring in economical feasibility and technical feasibility in the production of such high-value, low-volume molecules. So let me begin. If you see the slide, the topic today, what I wish to talk about is the motto of my lab. We develop plant and microbial biofactories for production of many of these high-value, low-volume phytochemicals. Now high value because they have a high market demand, and obviously the cost of these molecules is high. Low volume because in nature they are present in very low quantities, and that's why the gap between the demand and supply in the market is quite high. So our objective is, and many of these molecules are quite chemically complex in nature, so the chemical synthesis is either not possible, or even if it is possible, then it is quite complex in nature, which with less yields and also with maybe toxic byproducts release, or the biological activity required gets diluted because of the racemic mixtures coming out of those chemical reactions. Now with these demerits, now one of the key demerit to natural plant extraction, so conventionally these phytochemicals are extracted commercially using natural plant extraction. So we knew that many of these plants, to meet the industrial demands, were getting endangered. Now many of these, especially medicinal plants. So we thought, can we as bioprocess engineers, can we have an alternative to natural plant extraction for commercial production of these high value, low volume phytochemicals? So with that in mind, we thought, let's develop microbes and also plant cells and use them, mass scale them in reactors and then extract out the phytochemicals desired. So as I was talking, this is the motivation of the lab. If you see, 25% of the 150 top prescribed drugs, we cannot deny they are indeed from plant sources. The projection is that by 2050, the market of herbal products is going to reach $5 trillion. Very recent report which was displayed by the government on the Ayurveda Day says that there was a sharp increase in the exports of herbal products and Ayush products between 2013 to 2022. This increase was 67%, which is huge. That shows the potential, the current potential in the market for herbal drugs. Then 70% of the Indian medicinal plants, they are till date involved invasive methods of extraction. So which means that no wonder why increasingly many of our high value medicinal plants are getting endangered or on the verge of extinction. Now we know that most of these extraction processes involves natural plant as the biomass being sourced. There are key limitations that I did mention. The molecule being complex and chemical synthesis, sometimes it is not possible because the pathways are not deciphered yet. So the cues to do chemical synthesis is difficult. Now natural plant extraction, the problems is that many of these plants, when you talk to industry, you also get aware of the fact that they are facing the problem of adulteration, where the mixing of the key plant is being done by the vendors using the similar looking plants in the same variety. So that becomes very difficult for the industry to separate it. Now the other problem which they are facing is climatic and geographical variation in the phytochemical makeup of the plant. In fact, I'll be talking about a story on cyclotides which we did, and we in fact could see it ourselves that how geographical variation is bringing in variation in the cyclotide array in the plant itself. So this is a well-known fact. Now when an industry is dependent on a plant source for production of phytochemicals, it may be seasonal plant. Now knowing that in different geographies, because of climatic conditions, humidity, altitude, temperature, there can be a variation in the expression of these phytochemicals in the plant depending on the need. Now because of this, if you harvest the plant in a different season, you may see a different metabolite coming out, or you may see the same metabolite under different quantitative conditions. So it is not only qualitative variation, but it is also a quantitative variation which is observed. So now imagine how can an industry be dependent continuously on such a source which is so variable, inconsistent supply of not only the biomass but also of the phytochemical. So with these disadvantages, what we propose is, if you develop a plant cell or a microbial based bioprocess and you do mass cultivation in a controlled environment in reactors like you do in industrial biotechnology, then obviously you can continue to produce those desired phytochemicals in the same qualitative and quantitative manner, isn't it? Then the other advantage is you are no more dependent on the nature. So there is no geographical climatic variation. Other advantage to add to it is these cells are amenable to optimization, unlike the natural plants. So which means that you can manipulate the culture conditions so as to divert the metabolism of these cells majorly towards your desired product. Otherwise, the plant is producing plethora of secondary metabolites and other metabolites in nature. So the same carbon flux is being diverted towards so many different metabolites. And now you can connect the dots. No wonder why these are high value, low volume. Now if you want to fulfill the demand or have maximum productivities of a particular set of metabolites, you can manipulate the culture conditions, you can manipulate the cellular metabolism using metabolic engineering and or basically genetically modifying the cell so as to divert the major carbon flux towards the desired metabolite. So that is only possible when you are working in vitro, unlike natural plants. So that is why we got attracted towards developing these microbial and plant cell biofactories for producing these high value, low volume phytochemicals. Now once you have diverted the metabolic flux towards the desired metabolite, obviously then the downstream processing cost also can get reduced, because otherwise the plant is producing so many different metabolites, separation and purification becomes much more challenging. Now having said that, although developing microbial and plant biofactories have these distinct advantages over natural plant extraction, but there are still bottlenecks. So the idea is to develop strategies in order to overcome these bottlenecks to enhance the economical feasibility of these microbial based or plant based bioprocesses for production of these phytomolecules. So that is our motto of the lab. So these are the three pillars across which we work. We try to integrate all these three so as to develop rationally bioprocesses for production of these molecules in order to bring in enhanced economical and technical feasibility. So if you see the three key pillars, we are working on process optimization, which is manipulating the culture environment, the process parameters, where bioreactor design comes into the picture, where your environmental conditions optimization comes into the picture. Now, I would like to draw your attention towards the word rational. Now, why we are interested as engineers always to rationally optimize or rationally design? Because we would like to not only get to the target, but also economize the process of reaching to the target, which means in minimum time and in minimum cost. So what can be done in order to reduce this time of optimization, where we bring in methodologies like mathematical modeling, which I'm sure you all are aware of better than me, how applying mathematical tools to these biological systems, we can reduce the number of experiments, rather hit and trials, to reach to the optima of these process conditions, which are leading to maximum productivity levels. So that's what we do in that pillar. Now talking about the second pillar, which is metabolic engineering. Again, here what we do, now we move on from the culture environment to the inside cell environment. Now inside the cell, it is cell metabolism which is responsible for producing a particular desired metabolite. We also try to manipulate the cell by manipulating the enzymes present in the metabolism of the cells. Now manipulating the enzymes, in a sense, our target is overexpression of those enzymes or down regulation of those enzymes, rather knockout. So for this, what information we need to have? We must know the pathway, which is what reactions in the metabolism are responsible to reach to that product. Now in these reactions, which are those biological catalysts, which means enzymes, which are responsible to drive those reactions forward? What can be done to improve the activity of those enzymes so that the rate of reaction in the forward direction can be maximized, in simple terms? So generally what people do is, people increase the enzyme present in the cell by overexpression. People also knock out the enzyme if they find that it is getting diverted to some undesirable product, the carbon flux. They will cut down that part by knocking out the enzyme so that the carbon flux move towards, increased carbon fluxes moving towards the desired metabolite. So with that in mind, again rationalization, how do we bring in? One is metabolic engineering, that requires the pathway information. Now you can only tweak it intuitively if you have an idea about the pathway. Now imagine the cell is not only your pathway, but the cell has lot of many other metabolic reactions going on, which are catering to various secondary metabolites as well as the central carbon cycle. Now this primary metabolism will affect your growth, which is key. Now when I am trying to produce a particular product, I cannot just keep a focus on increasing that product without forgetting that the cell has to grow also. Only when I have an enough amount of biomass with the high amount of product per unit cell, I can think of having large amount of product in the end from that biomass. So the idea is always to keep a balance between these two. Anything which I do to improve the product should not bring in a reduction in my biomass growth rate. So I try to make a balance. How to make that? Because I am right now working intuitively. I am assuming that if I do overexpression in this pathway, which is very close to my desired product, so hopefully I will be getting high product, but I cannot guess what will happen, to what extent the biomass will come down. Basically when I am working with secondary metabolites, it's always an inverse relationship. So if you want to rationalize this and have the power of prediction before you experimentally validate that. Now especially in processes which involve plant cells, the growth rates are very slow. The batch processes, for one cycle of the growth to complete, it takes almost 10 to 15 days on an average. So imagine the time, the energy, the resources which will be spent to run one fermentation cycle, and that too a batch fermentation cycle. If you do some feeding in between to improve the productivities or the growth or the product, it can go to a month also. So if you do hit and trial, how many hit and trials and how much cost will it involve and time will it involve? So there comes the power of modeling. So in terms of metabolic engineering, we also do genome scale metabolic models, so as to have, in terms of metabolic reactions, the makeup of the cell, and then we do predictions and find out where the metabolism should be tweaked in terms of overexpression of the enzymes or knockout of the enzymes, and try to predict how much will it impact the product formation rate in terms of product flux. Now we not only do that, but we also rank the targets. We have found a strategy which I will be talking about, where we can rank those targets, predicted targets. We pick up the top ranked target because the probability of success will be highest. When anyways it has to be done, we imply some more constraints so as to pick up a target which will not only reduce the number of experiments, but which will also have the highest probability for maximum product flux. So that's what we do while rationalizing metabolic engineering strategies. Now coming to the third pillar, which is a novel and a very interesting and also very challenging pillar. Plant endophyte interactions. Let me give a brief introduction of what are endophytes. Endophytes are microbes which are residing in the plants. Now through evolution, there are so many microbes. It's very interesting. The plant allows one class of microbes to keep in itself. You grow within me. And one class of microbe it identifies as a pathogen and works against it, evades it. Now there has to be some mutual beneficial association going on between the two in order to the plant to keep it inside, allow it to grow within it. Now how can we exploit this under in vitro conditions? Now this mutual beneficial association, if it can be identified, so it cannot be one single way. It has to be a comprehensive association, which means that maybe it is helping the plant in its defense, also it is helping the plant in its development, in some maybe as an attractant to pollinators or to evade a particular pathogen. So it may be working together with the plant. How is it working? Through these phytomolecules. So what we figured out back in 2010 when I had joined the department, that it was a very recent paper we picked up from USB Bangalore, where they had published that they could identify a microbe which was producing a key plant metabolite, a very high value metabolite called camptothecin, and they deposited it in the national repository. That's what triggered and motivated us, but they could also identify the bottleneck there. They mentioned that, as reported by others in literature, that these microbes, although they seem to have the potential to produce the plant metabolite, but the minute you isolate them from the plant, you try to grow them independently under exinic state in the lab, the microbes gradually stops producing the plant metabolite. So that becomes the key limitation. You cannot be dependent on something which is inconsistent producer. So then we thought, can process optimization, this could be, what we hypothesized was, this could be related to the in planta stimuli or pressure on the microbe when it is in the plant. So something is happening inside the plant which triggers the cryptic machinery to keep producing that molecule, and maybe because under exinic state that is missing, because of that there is product yield attenuation observed. So we thought, can process optimization help us to bring in that stimulus back so as to revive the production of this plant molecule? So I'll be talking about that exploratory research also. So where we could get the lead, that was very interesting, and that again opened up the third pillar to keep following microbes also as one of the independent producers, alternative method of producing these phytomolecules. So with so many different things in mind, I am a bioprocess engineer, I cannot do it alone. I've always believed that you need a team to work on if you want to have impactful results. So we have developed until now a good ecosystem to help us to bring in those rational pillars. We are working as plant cell and microbial fermentation specialists. We have collaborators within the department, within IIT and also outside IITs who help us in biological activity analysis, because in the end you need to prove that what biological activity the natural plant has, you can also get from these in vitro cell lines. So biological activity, we have collaborators in biological sciences who help us to do mode of action, compare with the natural plant, with the commercial molecule, and prove that yes, it is equal. Then metabolic modeling, we have computational biologists on board who help us in doing genome scale metabolic modeling and first principle based kinetic process modeling. So wherein people like you who are experts in mathematics play a role. Then metabolomics, we have chemical biologists in the department, and also elsewhere we have collaborated who help us in qualitative and quantitative characterization of one or more phytochemicals from the biomass which we harvest from these in vitro cultures. So in the end you need to prove, if you are coming up as an alternative process to natural plant extraction, the first question comes to mind, is it similar or dissimilar? So then you need to prove that yes, the molecule which is coming out is similar to the nature. So there you will be needing a chemical biologist on board. Now what is more important as a process person, to bring in economics, there is a term called one organism many products. Now, many compounds or SMAC strategy. Now, when you can develop such biofactories which are producing more than one high value molecules, you can reduce the cost of production. So, for that, what is important? It is important to do metabolomics. It is important to know what are those array of metabolites which are getting produced under different conditions or under different stimuli from these biofactories. Then comes metabolic engineering, where sometimes we take help from genetic engineers who help us to engineer these plant cells. This is one of the successful leads, the initial ones. This was done by Aparajita. She is the first line of PhD students. She also helped in establishing the lab. So these are my strong pillars who helped us to move forward. She is right now in UC Berkeley. So what we did in this project was, I was mentioning about rationalization of cellular metabolism, the metabolic models. So we collaborated here with one of my colleagues, computational biologist, Professor Karthik Raman. So we developed a genome scale metabolic model for sunflower. The key molecule which we wanted to enhance was alpha tocopherol. Alpha tocopherol is the most bioactive component of vitamin E. Now that time we were thinking vitamin E is a good molecule to work with because it has a good market demand. So we picked up alpha tocopherol, and we could also find out that generally alpha tocopherol is extracted from seeds, and naturally seeds produce gamma tocopherol more than the alpha tocopherol. We figured out that sunflower by nature was driving large amount of flux towards alpha tocopherol. So we thought if we want to develop a plant cell based bioprocess, the starting material has to be a high yielding material. So we chose sunflower for that. Now the second thing was, even in sunflower, knowing that there is geographical variation, so if you will select different varieties, you will find there is different content of the product in all these different varieties. During my PhD research, we had collected 23 seed varieties of Azadirachta indica from the entire country. All these 23 varieties, 23 to 26, demonstrated different yield of azadirachtin. Azadirachtin is a biopesticide, very, very costly. So it's well known fact that with different varieties there will be different content. So we picked up, for getting a high yielding cell line, we picked up a hybrid variety, a commercial variety of sunflower from TNAU, and we figured out from 2 to 3 batches the average value of alpha tocopherol was nearly 160 to 180 microgram per gram. Now the idea was, after developing the cell lines, what we could find was, again as expected, there was a large dip in the alpha tocopherol yield. If you see on the slide, the seed has around, what we have shown here is 190 micrograms per gram. The cell line, which was a cell line from this variety, although commercial variety with high amount of alpha tocopherol expected, it was 18 to 24 microgram per gram, such huge dip. So how can you begin a development of a plant cell based bioprocess with such low yielding cell line? So then we thought, let's overexpress a particular enzyme in the pathway of vitamin E biosynthesis and try to generate higher yielding cell lines. Now to make this process more rational, the reasoning which I gave you some time back, we started looking if the sunflower model is already available in literature. Unfortunately, that time sunflower was not sequenced and the genome was not annotated, and there was no model available. That became our first bottleneck. So then we thought, how can we try to be much closer to the reality? So we figured out that Arabidopsis model was available. Now one can think that Arabidopsis is so far in phylogenetic relationship with sunflower, but there were existing certain logics based on which, like collinearity exists between distantly related plant species. Then we could also find that cDNA microarray of sunflower was constructed using Arabidopsis genes. So all this encouraged us, and theoretically we implied that to justify the adaptation of Arabidopsis model for sunflower. Although we adapted, but it is important to now tweak it towards sunflower so that the predictions can be real, close to the sunflower. To do that, we then figured out in literature, looked at reactions which were specific to sunflower. We added those reactions to the model. We did curation because modeling, that's the basic step. You need to do curation. So model was curated. Then we also could see that somebody had published 13C based flux map for a sunflower seed embryo. We thought, can we use these experimental flux data to further constrain the model so as to come up with a model which can be much closer to predicting sunflower metabolism. We did that using certain tools which is called as FSCOF, and then we simulated that model using FPA, flux balance analysis, to predict those enzyme targets in the entire metabolism of sunflower, which should be overexpressed in order to increase the flux of alpha tocopherol. Now please note that apart from alpha tocopherol, there are various other isomers of tocopherols which are called as tococremanols. So there is tocopherols and tocotrienols. Now our objective was to enhance the flux only towards alpha tocopherol. So we were wanting to predict those enzyme targets. The model was able to indeed predict some 23 targets for us in the entire metabolism. Some were very close to the pathway, so it could be easily predicted. So the question comes that could anyways be done if you know the pathway. So what's new about model? What we could also predict was some particular targets in chlorophyll metabolism which were coming up, and also in central carbon metabolism, which one can find very challenging or difficult to think that they might be impacting alpha tocopherol. But knowing that the plant cell, that is not our object. Many interesting leads were there. Fundamental sciences people could have taken it forward, but as process people working on time and optimization, we wanted to pick up a strategy which had highest probability of success and which could lead to maximum enhancement in alpha tocopherol. Now, rather than working on the novel leads, we then found a mathematical parameter. We formulated it. It was called as FPH parameter, known in literature, but we modified it because as a process engineer, I did mention that what is important, both to keep a balance between the growth and the product yield as well. There's no point having maximum product yield, which means amount of product in the cell, a cell which is not able to grow. So even in the modeling, we could find that if you keep on increasing the flux towards the product, the flux towards the biomass keep decreasing. So maximum alpha tocopherol flux was being produced at the point where biomass flux was 0. So you cannot have no growth rate with large amount of product, or minimal growth rate with large amount of product. There's no point. There'll be no cell to extract from. So with that in mind, we restricted the biomass tip, and we then ranked these targets for maximum alpha tocopherol production. We experimentally then picked up the top ranked target. We did the generation of cell line using agrobacterium transformation methods. So experimentally we picked up that, we validated that, and what nice results came out was, to our pleasant surprise, was that the cell line which we could generate after experimentation was a 10-fold higher cell line for alpha tocopherol than the control one. When we compared it with the natural seed yield, we were almost the double to the natural, so which was a very good news for us. So this I would term as one good successful platform technology which we could develop in the lab for producing high yielding plant cell line. Now with this work done, what I also realized was, and we as a group were discussing, if we could have done the same thing, spend the same energy, cost, resources on a product which was very, very high value, low volume, on a plant which was endangered, the impact could have been much more. So that's where we began the story of working on camptothecin, and now the current focus is on this molecule for the entire lab for all the 3 pillars, because it is being produced by a plant which is endangered. Sometimes there is a huge gap between the demand and supply. Almost if the annual demand is 3000 kgs, then the supply is just 600 kgs worldwide. So there is a huge demand for this molecule. If you see the CAGR value, it is close to 18% for this molecule. If you see literature, right now in the market there are almost 3 to 4 derivatives currently being used of the camptothecin is the lead molecule for irinotecan, topotecan, exatecan. So these are the drugs which are currently used in chemotherapy for various types of cancers relevant for our country: oral cancer, cervical cancer, small cell lung cancer. So this becomes a very good molecule to work with in terms of socio-economical impact. Now the other point is that this was being extracted commercially using the natural plants Nothapodites pneumoniana in the country, and this was identified for the first time from a Chinese tree, Camptotheca acuminata. Now both these trees, because of extensive uprooting, have got endangered. In fact, during my discussions with the industry as well as in the government, I could find that yes, whatever is said in the articles, that there is a strong depletion at a high rate for these plants to fulfill the market demand. So it becomes imperative to come up with alternative process of production for such a high value molecule whose demand is continuously increasing, and therefore the cost of the molecule is also continuously increasing, and no wonder the associated cost of the cancer treatment dependent on this molecule would also increase in the market. So if now the question becomes very relevant. So then we picked up this molecule and we extrapolated the same platform technology which we had developed on vitamin E on camptothecin. So we developed the cell lines of Nothapodites pneumoniana, and after applying the same platform technology, I'll not repeat it. This work was done by Saryu. She is currently writing thesis. Again, this was a very challenging tree to work with. It's a tree, so it's very, very challenging to do experiments with these, even to generate cell lines, and on top of that, to genetically modify them, to genetically engineer them is also very challenging, because to infect them with agrobacterium, a soil bacterium, they are themselves so toxic trees that it is very challenging to infect them with a bacteria, to transform them, to drive, to get the gene of interest inserted in the plant genome. So this was a very, very challenging work, and I'm pleased to say that Saryu has been able to bring it to a very good point, where we have now in hand the transformed cell line which is metabolically engineered and has been found to be producing 5-fold high camptothecin yield. There is still a lot of scope in improving the yield further, but yes, the proof of concept has been laid in this story also. So we could also predict some novel targets as we had done in vitamin E. We could see that amino acid metabolism and your TCA cycle was also impacting camptothecin production. Now the exploratory research which I was talking about, exploring microbes for production of molecules, we picked up camptothecin again. This work was started by my first MS student in 2011, where we picked up the same strain because it was available in the national repository, which was deposited by UASB. I told you that the group had first time reported that they were able to find a microbe which was producing camptothecin from the Indian variety. We took that strain, and I would say this was a very serendipitous discovery, where we were thinking that culture manipulation, the condition manipulation, should be able to change the product yield, camptothecin yield in the culture. With that in mind, we were working with environmental conditions optimization, then medium composition, and also then we thought, let's also work with well-known strategies known in plant cell fermentations where we add certain molecules. One are the class of molecules which are intermediates in the biosynthetic pathway of that product. So assuming that if the molecule can be readily made available to the reaction center in the cell, the rate of forward reaction would increase because you are increasing the substrate availability. So with that in mind, these are called as precursors. That also led to some enhancement. Another most promising strategy still date in plant cell fermentations is addition of those molecules in the culture medium which gives the signals to the plants that something wrong is happening, which triggers their defense mechanism. And if the molecule of interest is a defense metabolite, most of the times these secondary metabolites, they get enhanced because of this cascade effect in the secondary metabolism of the plant cell. Now with that, we thought that let's extrapolate it to the endophyte as well. So we picked up the similar which were impacting the plant cells, the same elicitors, and we had put it in the medium. We saw that camptothecin production from this culture, which was very, very low yielding culture, nanograms per gram, was also showing some bit of enhancement. Now one interesting experiment came to mind. With Aarti then came and discussed with me that, ma'am, can we add strictocidine? Strictocidine is an intermediate in this pathway of monoindole alkaloids. After strictocidine, it gets diverted to many different kinds of alkaloids. One of them is camptothecin. And after strictocidine, the type of alkaloid, the kind of plant is also different. But till strictocidine, monoindole alkaloid pathway seems to be common. So then I said, okay, let's buy strictocidine and let's try to add it and see if camptothecin production improves. We checked who can sell strictocidine. Strictocidine was not available in any of the vendors known to us. Suddenly she said, ma'am, a Chinese vendor is ready to sell it, and we also asked that, okay, let's get the molecule, but somehow the PO was released and that could not materialize due to some limitations. So then we thought how to now take it forward. Then an idea came to mind that Catharanthus roseus grows wild in the campus, and Catharanthus roseus also produces vincristine, vinblastine, which is also an indole alkaloid, monoindole alkaloid. So the idea came that till strictocidine the pathway is common, so why not take some extract? Let's see what kind of molecule strictocidine is, prepare the extract assuming that strictocidine would come into that solvent system, and we'll add that extract and see what happens. So she did that experiment, and she came running to me after she had done the analysis that, ma'am, it has shown very high enhancement. I said, very good, repeat the experiment, let's see what you did. So by mistake she had forgotten to keep the control, the solvent system. I said, no, this will not sell until unless you have the solvent also as a control in the experiment. Go back and repeat it. So she said, yeah, that's a mistake, let me go back and repeat it. She repeated, she came back more higher jump. So she said, ma'am, the enhancement has increased 15 folds. I said, that's wonderful. You need to make a check 10-15 times before you come to me. So then she again went back, she did the same experiment 10 to 15, because I was also nervous, she was also nervous, I was equally nervous that how come 15 fold is too high to believe, let's try it again. So she tried, she had done the experiment, I think I'm telling you 15 times, we actually did it many times because we really wanted to be sure. Wonderfully well, every time the experiment worked. The culprit was the small molecule. The control was showing the maximum enhancement. So we said, let's do one thing. With the graded concentration of this small molecule, let's see what impact is it playing on camptothecin. So we could see that with the graded concentration, there was a proportional increase till a point where biomass was also increasing. This is unlike elicitors. Elicitors, because these are defense-related molecules, they give a signal of something wrong to the cells. So generally what is observed is that the biomass growth, the growth rate reduces, and although the product yield improves. So that was not expected. So now the next question was in mind, how come this small molecule is having a good impact on a secondary metabolite and also helping the cell to grow. That's not expected. So let's do one thing, let's find out the role of this small molecule. So this was an MS work, just 2 years work, but she did a lot of, she was I think one of the best in that lot, and she designed an experiment where we could find out that the total protein content was also improving, and this molecule was being used by the cell as an alternative carbon source. And we also did protein analysis. Although we could not get in deeper, she had to submit thesis, and again repeating, as a process engineer I can only go to some extent, but then I need to come back because the objective is to maximize productivity. So then, but that lead could have been forwarded or could have taken in depth, where what is the role of that small molecule. We patented this work. We recently got this patent granted on the small molecule, which has led to enhancement of a plant metabolite in a microbe. So this strategy can be generalized to many such plant metabolites to see what impact on enhancement most probably it can bring to other endophytes also, producing other plant metabolites in alkaloid biosynthesis. So that has to be checked, but nonetheless the strategy could lead to maximum enhancement in camptothecin production, which could also lay the proof of concept that yes, microbes can be sustainable producers of plant metabolite, but the key lies in the fact what environment is being exposed, where the culture is exposed to, in order to keep producing that metabolite in a sustainable manner. So now once the concept was laid, what we also then realized was that we cannot be dependent on this cell line. One, it is not ours, it is in the public repository. The process, it is a very low yielding strain, so developing a process to bring in economical feasibility is not possible. So it was a nanogram yielding strain. So putting up lot of resources, time, energy on a very low yielding strain is not logical. So then with the proof of concept laid, Khwaja, who had then entered the lab as a PhD student, we thought of going back to nature and do some bioprospecting. Let's bring in a microbe which is very high yielding camptothecin to begin with, and then implement these optimization strategies to make it economically feasible. That was our goal. So Khwaja, this was a collaborative study done along with the group in UASP and those who had isolated the first strain. So they helped us to get the plant material, because this is endangered. You cannot just go in the wild and even for research extract it and start isolating the microbes. So you need permissions. So then we collaborated with them, we got the material, he isolated almost more than 100 endophytes, he then screened them both qualitatively and quantitatively. More than 90 of them were found to be producing camptothecin. We confirmed it using HPLC, using tandem mass spectrometry. We then ranked them. We selected the top 6 ranked endophytes, and now the key for success was to again do a stability analysis of these strains, because even though we had top 2 strains which were highest reported till date on the camptothecin yield, so we were very happy about that, and this was very close to the average yield in plants. So that was a good news for us. But then the next bottleneck was that will they continue to produce the same metabolite in the exonic state or not? Now what if you begin a reactor and after 100 generations the product is lost? No industry can work on an inconsistent producer. So then we thought of selecting those top 2 microbes, and we did a stability check. Generation wise, this was a continuous study, I think more than 3 years. He kept on subculturing. The batch period was of 8 days, and we kept on seeing the dip in the camptothecin yield. Now another interesting fact which came, I think, how much time do I have? See, as a scientist, when you begin talking, you don't realize where the time flies off. Okay, I'll just wind up very quickly. So then what happened that we picked up the top 2 strains and we did that sustainability check, and we could find that the second highest yielding strain was showing an asymptotic behavior. If you see that, is this a pointer? Yeah. This graph, this is the graph for the second highest yielding strain. The highest yielding strain soon after the second subculture showed us very close to that x-axis. So that was in alignment with what was reported in literature. But the second highest yielding strain for more than 100 generations was showing us an asymptotic behavior. It stabilized near 200 microgram per gram, which was still the highest reported in literature. So we thought this can be a good candidate to take forward for process optimization. Now during this bioprospecting, one interesting thing which Khwaja could find out and develop was a rapid screening methodology for rational bioprospecting of high yielding endophytes. What he could find was that the highest yielding endophytes were coming from high yielding explants, plant parts. So plant is also producing the molecule, the microbe residing in the plant is also producing the microbe, that molecule. So when we did the analysis, we could see that the higher yielding endophytes among the 90 which were producing camptothecin were coming from high yielding explants. And another interesting thing which he could find was that the higher yielding endophytes were demonstrating higher tolerance towards camptothecin. Camptothecin is a cytotoxic molecule. So were demonstrating higher tolerance towards camptothecin than the non-producers of camptothecin. So which can be used as a rapid screening methodology now, the tolerance towards camptothecin, when you want to bioprospect high yielding endophytes for camptothecin. So that was another thing. Now in order to prove, when we were digging literature, we could also find that the group which doesn't believe that endophytes can in fact produce phytochemicals, host plant associated chemicals, say that it is not due to the endogenous metabolism that this molecule is being produced in the microbe, but it is probably because the molecule is being stored by the plant in these microbes, that when you isolate them, you in the initial generations you see the molecule in these cultures, and that's the reason why it fades off. Now if that is so, you would not see sustainable production, one thing. Now how to prove, our next objective, what Khwaja did was to prove that indeed this molecule was coming from the metabolism of the cell. So we did 13C labeled metabolite analysis. We took the starved culture, we exposed it to fully labeled glucose, and when we grew the culture, the camptothecin, which is an intracellular metabolite, we extracted camptothecin from the culture and we could see it was labeled. So that established the fact that camptothecin is indeed coming from the cellular metabolism of this microbe. So we are now at a stage where we have in hand a microbe which is producing this high value molecule. If we can take this forward, this would be an indigenous technology which can also substitute camptothecin import in the country. We can even establish ourselves as camptothecin manufacturers, knowing that the molecule is high in demand worldwide. So being alignment with all what is being demanded today. So these are the advantages. These are all those advantages which I spoke to you, which a plant cell fermentation or a microbial fermentation can have over natural plant extraction. This is the journey. We have been able to develop plant cell biofactories for producing camptothecin. We have been able to develop microbial biofactories for camptothecin. There has been a good visibility for the work which we have published that also demonstrates in some way the socio-economic potential which the work can have. And there are n number of other small projects which we are continuing now, which we want to do in collaboration. We are doing it to take the technology forward. We are currently at TRL 3 to 4 level and we wish to take this forward to TRL 8 to 9. I'll close here. This is another work, cyclotides. We as a group were the first ones to discover cyclotides in the country using Fourier transform mass spectrometry based analysis in the Indian plant variety of Banafsha, which is Viola odorata. So this work was done by Narayani, and then this work was taken forward by Babu, another PhD student, where we picked up that cell line, and that cell line from which is capable of producing cyclotides and other molecules, we are taking it to the reactor level. He is a PMCII fellow, and with the industry partner, he is now going to the industry to reproduce that result, and we are hopeful that something good can be brought out for commercialization. I would like to thank my group. It is not at all possible. I'm sure everybody here realizes that until unless we have motivated researchers on board, the work cannot move forward. So this is my group, some past and present students. These are my collaborators. Again, as I told you, that rich ecosystem which we have developed, this also has helped to make the wholesome stories. This is the glimpse of my lab. These are the different types of plant cell cultures which we work with. Hairy root cultures. These are my students working on the bench. This is the photos of my lab. These are plant cells growing in the reactor. The bottom one is the fungal fermentation. Thank you for your attention, and I hope I did justice. Thank you.
So Smita has a short presentation about the bioincubator in IITM Research Park, few minutes, and then you can ask questions.
Nothing is coming out. Can someone help? I did presentation view. Is that the problem? You can see. I said no presenter view. Is that the problem? So escape, huh? I'll close it. I'll open it again. Yeah. Does it come up? Yeah, I'll just take maybe 10 more minutes to be true. Okay, so I also happen to be the faculty in charge of IIT Madras Bioincubator. IIT Madras Bioincubator is stationed in IIT Madras Research Park. This is the first university-driven research park. IIT Madras happens to be the pioneer in it. And IIT Madras Bioincubator is a place where we want to nurture bio-entrepreneurs. By providing them a holistic environment, a platform where there can be not only technology advancement taking place, but in parallel for commercialization, there are business aspects, financial aspects, which also cannot be ignored. And most of the times, knowing that biotech domain is a long drawn process, it takes time to reach to market. Most of us, when we want to become as entrepreneurs, we try to focus on developing the POC first and then think about finances, but actually that's not the case. One has to keep financial model, the business model equally at the same pace. You need to develop and focus on it as you would want to focus on technology advancement. So we try to make them aware of what are the needs or what are those prerequisites, and we provide them legal support, IP support. So bioincubator sitting in research park ecosystem provides that advantage. There are various domains to which we are currently catering to. We have agri-biotech companies, we have industrial biotech, that's our forte, industrial biotechnology. Then we also have companies in computational biology domain. We have some pass outs in veterinary domain. We have environmental biotech startups and also therapeutics and diagnostics. So this is a 10,000 square feet facility in Research Park. We also have a 700 square feet facility sitting in the department, which is meant for pre-incubatees. Now pre-incubatees are those where we see the potential for becoming entrepreneurs, and we do hand holding at the ideation stage itself. Sometimes idea may be good, but it may not be commercially feasible. So we help them to rework on these ideas by rigorous involvement of certain programs conducted by IIT Madras, Entrepreneurship Cell and Gopal Das Center, where we do customer discovery. There is vetting of your idea, how commercially attractive it is to further take it forward as a startup. So this is the glimpse of the facility. The first column shows the department facility, mainly for pre-incubation. Sitting area, we also have meeting rooms, conference facilities to help the startups. Then apart from the equipment infrastructure, where we have more than 100 equipments, it's a wet lab facility. We cater to deep tech startups. So we do not have large number of companies, but whatever we have, there is a thorough screening process by which we induct incubatees and bioincubator. We provide them with networks which we have developed with various state level agencies and also National BIRAC authority for funding support, which we can provide initially to these startups. Then in the 10,000 square feet facility in Research Park, we have one co-working space, the middle column, where we have the infrastructure, equipment infrastructure in the open area, and the sitting area which we provide as an office space is also a co-working space. Now in phase 2, if you see the last column, it is an exclusive facility where we provide cubicles. These are nearly 160 to 180 square feet cubicles which we provide to these startups, which can be customized as per their need. And the common labs are open to all. So in general, if you see, then it is nearly 500 to 700 square feet of space per incubatee which we provide. And this works on a rental model, let me state that. Then the different kinds of high-end facilities which we have, they cater to cell culture, microbiology, synthetic biology, analytical facility. So these are in fermentation, of course. So we have RT-PCRs, we have GC, we have HPLC, we have FPLC. These are all high cost equipments which we provide to our incubatees. Otherwise, as startups working in biotech domain, it's quite a capital intensive domain. So providing such a comprehensive equipment infrastructure is the key which is provided to them. And in fact, if you see, even though we work on a rental model, almost 60% subsidy which we provide to our incubatees. So this is a BIRAC BioNest incubator running since 2014. This is a pilot scale reactor. Fermentation is our forte there, industrial biotechnology, although to other domains also we cater to. This is a very comprehensive, systematic way of screening incubatees. As I said, we want deep tech startups. This is one of the ways of reaching out to those incubatees, where there is an online application process followed by a pre-screening. Internal BI team is there who talks to these people to find out their idea and see how commercially feasible it is. You can see these are the generic criteria based on which we try to incubate. We look at the idea, we look at their business plan. Of course, team is very important. Then feasibility in incubation, how much can we help them? So when we incubate any startups, it is not only brand for which they might be wanting to associate with us, but we also see how much they need us in terms of equipment infrastructure which we can provide them, and also in terms of the rich ecosystem in Research Park which can be leveraged. So this is the process to market which we generally follow. All the incubatees, we begin with pre-incubation. If there are any potential entrepreneurs where we find that they are very motivated and enthusiastic, I would like to mention here one of our incubatees who were just college BTechs. very fresh out from completing their degree, initially came with an idea in this pre-incubation screening where they presented that they wanted to make an impact in environment cleaning. So, but we then soon made them realize that it's good to have that thinking that yes, you want to make a difference in terms of social impact, but it is equally important to have economical impact if you want to run a company. So your idea should also have a commercial potential. So then we made them realize through a series of training processes, and now I am very happy to inform they have finally incubated. The idea is now formalized to a very different. They are working on waste to value, where they are working on, I cannot disclose much, but yes, the idea is where they are working on mushroom based packing material technology, which is a replacement to styrofoam. So finally, after these many different processes they have gone through, and they have been able to generate close to 1 crore of funding also. So that's what the advantage of pre-incubation and hand holding is when you want to develop entrepreneurs. So you see and you feel very nice when you see these budding entrepreneurs maturing as they go to the market and becoming successful unicorns. So these are the ways in which we provide the support. As I mentioned, we provide legal support, we provide IP support through IIT Madras Incubation Cell. So we work hand in hand with IIT Madras Incubation Cell, which is an entity where all the startups have to be registered. So the agreements are signed with IIT Madras Incubation Cell, which holds the responsibility of providing the support of legal, IP, making a connect with the alumni of IIT, making a connect with venture capitals, making a connect with angel investors, because funding is important, especially when capital intensive domains like biotechnology is being looked into. And this is how IIT MBI is IIT Madras Bioincubator. There is a comprehensive support which we provide in terms of technology development. So the association of, this is our report card, possibly. So the number of startups which we have supported until now, these are 21. The jobs which we have created, more than 100. IPs which we have generated by our incubatees, more than 20. We have 9 already graduated. 2 of them are now generating revenue more than 1 crore. We have 8 products in the market. Funds which our incubatees during the incubation have generated is more than 30 crores. These are both government and private funding. And we also, apart from that, as our capacity building initiative, we also hold various programs to keep searching for budding entrepreneurs. So for that we hold webinars, we hold hands-on training programs. So as you can see, until now we have been able to hold close to 40 such programs. These are some of the event photographs. Yeah. The advantage of IIT Madras Bioincubator is that it is sitting in an ecosystem which is a bridge between the outside world and IIT Madras. So all these startups are exposed to, we become a bridge between the Department of Biotechnology as well as the research park ecosystem, where Bioincubator has IIT Madras faculty as founders, as mentors, as advisors, and many of the students are working as interns in these startups. They are also as founders in these startups. So there is an inherent connect between IIT Madras and IIT Madras incubatees, bioincubator incubatees. Yeah, these are some of the awards which we have received. Thank you. And any questions, please feel free.
Yeah, questions? On the science and also the bioincubator. Yeah.
If you consider plant cells from different parts of the plant, then so the phytochemical profile will change. So like how do you decide like which part of the plant you should go ahead with?
Very good question. So as I said, rational bioprospecting is also important. When we are developing high yielding cell lines, if you remember, I mentioned that we went back to 23 to 26 seed varieties of Aza directa indica, neem seeds. We collected those seeds from different parts of the country. Why did we do that exercise? The reason was we know that the kind of variety with which you will begin the work will have an impact on the yield of the cell line. So it is very, very important to quantify the yield of your desired product in the plant part which you are planning to use to isolate the cell line. So we did that screening. Initial screening is important. You quantify how is the product yield in these different plant parts, pick up the highest yielding plant part and take out the cell line, isolate the cell line, generate cell line from that plant part, and that will show you the maximum yield possible, and then work on optimizing the yield further for maximum multifold enhancements. Yeah.
Questions? Vivek?
So, in the second work on camptothecin from endophytes,
Yeah.
the first work was introduction of small molecule increase the multiple folds.
Correct.
Have you tried trying the same small molecule in the second study?
Right. So now that we have the strain in hand, we are developing strategies. Now what is important is not only the type of small molecule, but the concentration of the small molecule is also important. So that work is underway, and in fact second patent is also filed, and it is we have just filed the FER.
Questions? Any further questions? Okay. If not, then I had one question. So is the microbe a bacteria or a fungi?
It's a fungi. So although there are endophytic bacteria also, but we are working on endophytic fungi.
Now the related question I have is that when you did the initial work which Aparajita did, so did the metabolic model of Arabidopsis had well developed secondary metabolic pathways such that you could actually have those, or you built those?
No, we built.
Yeah, that's what I thought.
See, vitamin E is common. So you can find out reactions, and some reactions we had to add. So that comes under curation of the model.
Yes.
So the model was only made useful when we added the reactions. I think we added almost, if I'm right, 23 reactions. I don't exactly remember, but there were some reactions which were added. Biomass reaction was missing. We worked on the biomass reaction. So on the secondary metabolite front, like the work which was done by Saryu, now when we were developing from scratch the model, then from the sequence of Camptotheca acuminata. So that model we call as CamGem. So I'm happy to say that we have named it as CamGem S because Saryu had developed that model, and we are going to publish it that way. So in that model we had to integrate reactions of camptothecin which are already known in literature. So that work is required to be done in modeling.
So, so to be very precise, you have not found any ready resource to get secondary metabolism.
It is, yeah, it is
phytochemicals,
right. It is difficult because different plants will have different metabolites getting produced, and for example, you will find in literature that a plant is like, let's say, monoindole alkaloids. If suppose I am working on Nothapotites pneumoniana, which is camptothecin producing, and the model is available for vincristine, vinblastine, so I can find reactions up till strictocidine. So I will have to see similarity of enzymes which is between the two plants, and based on some logic, generally more than 40% similar, so we picked up, we adapted that, and those reactions remaining which are published in literature, we then add on to the model, and then we try to develop one model and we bring it closer to experimental, our own cell line. So there can be variation not only in plant to plant but even with the cell line. So that is where you need to constrain the model based on your experimental values. So what I mean to say is, working hand in hand with experiments and modeling is important. Only developing a model and then predicting it may not work. So you need to keep curating the model. It's a back and forth process. So you need to keep developing experimental data, feed to the model, and bring it, it's an iterative process, bring it closer to the right predictions.
Any questions? By the way, the endophyte is not sequenced, I'm showing.
We have done it.
You have done.
Yeah. So now the idea, let me just tell a little more about it. Once we had the endophyte in hand and we have the platform technology of genome scale metabolic modeling led metabolic engineering, we are also now, we have done the genome sequence, we are in collaboration developing a genome scale metabolic model for that organism in order to again extrapolate the same platform technology on this fungi to develop metabolic engineering strategies to further enhance the yield in the fungi.
That's awesome. I think there are no other questions. So let's thank Smita again for this wonderful talk.
Thank you. My pleasure.